Good morning. My name is Eileen Hoffman. I'm president of the Ocean Sciences section, and I'd like to welcome you to the Rachel Carson lecture. As a bit of an introduction, for those who might be interested, Rachel Carson was a marine biologist, and she wrote extensively about ocean life from the shoreline to the deep ocean. And she's perhaps best known for her book Silent Spring, which was published in 1962. And that book examined and documented environmental damage caused by widespread use of chemical pesticides. And that book was important because it produced enhanced public awareness of the environment and set up the environmental movement that eventually led to the formation of the Environmental Protection Agency. In recognition of Rachel Carson's contributions, the Ocean Sciences section established the Carson Lecture in 1998. The lecture is presented annually at the fall meeting by a woman honoree from the Ocean Sciences section. The past honorees were distinguished scientists whose research achievements continue the legacy of environmental research excellence. Today, we continue this Carson Lecture distinguished honorees with Claire Paris, who is a professor at the Rosenstiel School of Marine and Atmospheric Sciences at the University of Miami. Claire is a biological oceanographer with a diverse background in larval ecology, coastal oceanography, and numerical modeling. She is known for her research into the role of behavior of the pelagic larval stage in the connectivity of marine populations and the function of ecosystems. Her research focuses on biophysical dispersion at sea, as well as the transport and fate of pollutants, debris, and oil spills. Claire has developed numerical and empirical tools to support larval connectivity and transport research, one of which is the Connectivity Modeling System, which is an open source software system that allows you to virtually track biotic and abiotic particles in the ocean. She also developed the Drifting In Situ Chamber, which is a field instrument to track larval movement, to track movement and behavior of early life history stages. For these many accomplishments, the Ocean Sciences section is proud to recognize Claire as the 2018 Rachel Carson Lecturer. So I now invite Claire to give the Rachel Carson Lecture on Larval Odyssey and Marine Population Connectivity Now and Tomorrow.
Thank you for the introduction, and thank you for being here today. I am really grateful for the nomination and to the AGU Award Committee. I would like to share this award with my current team and colleagues, and also all my mentors and friends, students, postdoc, technicians along the years that continue to challenge themselves and work hard to better understand and conserve our ocean. I'd like to thank also my husband and my children for giving me strength and love. So I am deeply honored and to be here and humbled. Rachel Carson, in the words of Carson, I don't know, this is not the first slide. Okay. So this is my team, my current team: Nathalie Perling, Anna Vaz, Veronica Luchissé, Igal Berenstein, Robin Fayotaz, Atira Prasad, and Andy Cow, who I'm still working with. So a few words about Rachel Carson, really my hero. In the word of Carson, she was the best of America. She was a modest person, concerned, courageous, and profoundly right all at the same time. So I know I was influenced all my life by her because I was born around the time she was writing her books. And she had a big impact on the US, but also in Europe and all over the world. She was always at the edge, pushing further her scientific knowledge about the ocean and the natural world. Her attention to details was what I admire the most. Small creature, she described small creature as a naturalist, and it was extraordinary. So being close to nature gives you some intuition and give her the vision of the future. As a founder of the modern environmental movement, her work seems more relevant today than ever. So I will come back to this in the second part of the lecture. The AGU centennial focus on sustainability of the future, and this is in the same line of vision as Rachel's vision. Her history, and also is pertinent to the history of fish larvae with the theory of critical periods that just started with Yort in 1914. So today I will talk about the concept of larval dispersal from this theory of critical period to what we know today from testing the theory with in situ technology and numerical modeling. And the second part of the lecture will address the new threats to the pelagic environment and the challenge we face to ensure sustainability of our oceans. So what is dispersal? So in the broader sense of the term, dispersal is the movement away from the birthplace and is a common phenomenon that occurs in most living organisms at some point of their life history. For most coastal and benthic marine species, the only opportunity to disperse is during the larval phase. So climate change is already altering the pattern of the organism movement, yet we don't know the full spectrum of dispersal drivers in the ocean, and this is because we've seen always this larvae as passive. But what is a larva? So all fish, even those that grow to be big like the mola mola, start their life as microscopic larvae. When you look at them with detailed eye, you start appreciating the sophistication of their pelagic adaptation. In this case, they have elongate serrated spine that may help survive cannibalistic predators. And they're equipped with impressive sensory apparatus. You can see the size of the eye on this larvae is much larger in comparison to the adults. So that probably helps them sense the environmental cues. Another example is a squirrel fish, and this is the larvae. And what is intriguing is not only the beautiful of this form of life, but also what fascinates me is the abilities that I will describe later. So looking back at 100 years ago more, your critical period provide a foundation of the research that has generated a broad understanding of cause and variability in fishery recruitment. So according to this period, the critical period, the larvae survive depend on whether or not they find food when they are at the feeding stages. So this is this yolk sac larvae that has exhausted its reserve and now needs to find some food. So another second part of this hypothesis is that during this period and further on, there is also an aberrant drift hypothesis that was stated in 1926 by Yort, where basically it was only due to the current, the survival of the larvae were due to the current that were either favorable or unfavorable to the retention near shore where they could find the nursery areas. So there was a series of hypotheses, but if you look at all the hypotheses that were put up for understanding the variability of York class strengths in fisheries, basically they all were based on the fact that these larvae were passive in the current. So all the major hypotheses were taking into account the spawning grounds, but not the larval behavior. So to understand what the larvae are doing, you need to observe them. One of the most current technology to do this is an optical system, like the ectoplankton, the in situ ectoplankton study with the ISIS instrument. It gives the opportunity to see a synoptic view of this larvae. So you can test the hypothesis whether or not these larvae are passive and will aggregate in the frontal zone. Here we see that the black dots are fish larvae, and this is a Ligurian current in the Mediterranean. There are no larvae in the frontal zone, and they're restricted to the coastal zone. In addition to that, they have vertical migration during the day and the night, where during the night they're found near the surface, and during the day they're a little bit deeper. And this is the first time it's been seen because it was a behavior that it was eluded by the technique that we were using for ectoplankton study, because with these methods you can see the entire water column synoptically, while with other instruments you have stratified sampling. The only problem with this, which is a huge problem, is that the identification of the larvae through this system is not straightforward. And so far we can only see it's a fish larvae, but we don't know what the species is. So we go back to the traditional ectoplankton studies, where we use a mock nest net, which is a multiple opening and closing net environmental system that we capture the larvae. And we have to identify those larvae. So I spent quite a long time in my career to do some larval taxonomy, and that's how, like, knowing a little bit more of this larvae, I was more interested in what environment they were living and how they survived this pelagic environment. So what we see here, different larval patches from early to late stage larvae on 20-meter strata of the water column. This was done west of Barbados, an isolated island where we could test the retention mechanism because there was no upstream source of this larvae. So what we see that during ontogeny, so during the development of those larvae, there is a migration towards deeper zones. And basically that's what we were seeing also in the optical system. We see like the extension to deeper water for during the day. So how this retention mechanism works, basically is the interaction of the ontogenetic vertical migration with stratified currents. On the left you see the zonal and meridional current. In Barbados you have really strong offshore current in the surface, and there is current reversal from the north to the south at depth. At depth also there is onshore flow, and the flow is has less velocity than at the surface. So what happened in the retention graph that you see on the right side, you lose a lot of larvae at the beginning until day 6, where they actually are retained at almost 100%. And so the average retention is about 25%. And these 6 days where this occur correspond to the flexion stage of the larvae, where they start the vertical migration. So basically, you can see that even though this is a mean here, there is a lack of larvae in this area, but you have like two modes. You have either larvae that are retained at very high percentage, or larvae that are very poorly retained, like at 10% or less. So that confirms a little bit the aberrant advection, but this is nuanced by the fact that it only happens during the early life history, during the early stages of these larvae. Once they start migrating vertically in the water column, they're not advected by the currents anymore. So it's a revised hypothesis for the aberrant drift. So now we've seen that in Barbados, which is an isolated island, those larvae can be retained. But what happened to those larvae that are moved away from the island? So we go back to the Mediterranean here, and we looked at the connectivity between different marine reserve areas. And what we mean by connectivity is the larval fluxes, because the adults usually have a very limited migration, so they're mostly sedentary. But like I said before, the large fluxes occur at the larval phase. So we want to understand this connectivity to understand a little bit better how to place those marine reserves. So connectivity is important for the demographic and avoid risk of extinction. Genetic connectivity increases the genetic diversity, and also you can find the distribution and the resilience to disturbance of the species. It has also application for conservation, for fisheries, and also to anticipate the migratory pattern during climate change. So the theory of population connectivity comes from the terrestrial ecology and emerged during the beginning of this century. So now I wanted to show you a little bit more about the larvae. It's my passion for the mysterious worlds of the fish larvae that brought me to learn more about the physical environment. Here you see a group of late-stage larvae that we're following. This is taking place in the Great Barrier Reef. As you see, they kind of like group together, like flocking like birds, and they actually swim directionally. So fish larvae are skilled swimmers and work together to find their home. Again, this is the late stage where they are ready to settle and they're competent. So this lasts for 10 minutes, so I'm just gonna skip this and talk about their swimming speed. This was measured in flume by Robin Fayotaz for his dissertation. So historically, the mortality of the early life history is supposed to be due to poor feeding. But here, by measuring the critical swimming speed at the settlement stage, we see that they're a very fast swimmer. They swim on average faster than the average current. And when you transform their swimming speed in body length, you have two groups. One group that is swimming very fast, and one group is swimming like average speed. But still, when you do a comparison in body length, compared to fastest human, the fastest fish, the adult fish, swim 15 times faster than the faster human. And the chromis, the second here in the graph, swim twice as fast in body length than the adult fish. Faster car in comparison is less than that in body length. So that's, to me, they're the hummingbird of the sea, because they flutter the caudal peduncle, as you saw in the video, and they have probably a very high metabolism, and they can sustain the swimming as far as they're eating, and actually they're eating while they're swimming. So, but this happened also at the preflection stages, and we did some work in Austevoll in Norway, where we looked at the in situ swimming of larvae that were hatched in captivity, and we put them in in the disc instrument that I will show you later, and we saw that they have some swimming speed, and they're directional, and the swimming speed increase with ontogeny. So it's not only a trait of the late stage, it's a trait of the larvae at all times. So, but when you do, I don't know if you know about Orienteering, but it's a game where you have to run and orient at the same time with what you equip with is a map and a compass. And the winner is the one that can do this, find its way faster. So basically what I'm saying here is that larvae may have a map and a compass, because if they're swimming fast, they should know where they're going. So to test this, we use a numerical model, like the connectivity modeling system. Here is an experiment in the Great Barrier Reef with a very high resolution model, but you can see those particles that are settling on the green ones, and some are advected on the red one. These models have basically three modules that are working in parallel. The seascape module, which is a habitat, the oceanographic module, which is what you see in the background here, the 1 kilometer resolution model for the for Belize that was just out of the oven, I would say. It's Milan Kursik that this is modeling at Rasmus. And then you have the biological module, all feed into the larval dispersal. What we add to this is orientation towards the coast. And this, we did this in, again, in the Mediterranean, where we use the Mars 3, which is really fine resolution, enough to represent the mesoscale eddies and the circulation there of the Ligurian current. So what we did here, we had larvae that were deployed in all the grids of the model. They were deployed only 5 days before their settlement period. And we looked at the trajectories that stay offshore and those that settle and calculate the percentage. When we looked at that with different swimming speed, there are three different things that we find. So the directional swimming strongly increase the number of settlers. Second, it's a little bit independent from the mesoscale circulation that is here that would retain the larvae according to the aberrant drift or the retention hypothesis from Yort. And third, passive larvae can reach the coast from 5 kilometers from like pure stochastic drift, but the fastest swimmer can reach the coast in 5 days from 56 kilometers, which is very significant. So we looked at connectivity from the 3 marine reserves that are deployed in this region of the southern France for 2 different species, and we look at the passive scenario versus the active scenario where they were swimming on the coast. What we see is that the larval flux are enhanced by this behavior. What I want to point out also is that small Spiridae have a shorter larval duration, and we always say larval duration is driving the connectivity, but really the large Spiridae have a longer larval duration, and still the connection are lessened. So this is counterintuitive, but the point is here that with those behavior, you really increase this network. So we didn't draw any connectivity matrix for the Mediterranean, but to tell you a little bit about those matrices, they built based on location, so you have the source and the sink location. And this is very powerful because it quantifies not only the source and the sink, but also the potential of settlement probability from different places. And these matrices can be built on any time scale. They can be built on a daily basis or monthly basis, annual basis, and you can use this to calculate again the connectivity network and see whether you have nodes that are essential to maintain the network, and this is very useful for conservation, but also to look at the effect of climate change and other disturbance to the system. So if these are just modeling where we have observations, so those larvae are swimming, they're swimming fast. If they swim fast, they can be coming to the shore easily. But what are the compass of these larvae? So there is different cues in the marine environment that can help them navigate. And navigation, animal navigation, is something that has been very well studied for terrestrial organisms, but we fall behind in the for marine organisms. So this navigation skill can be driven either by the acoustic field, but it's mostly at the short range. Because those larvae are very small, they can only sense the particle motion and not release long wave signals. And you have also large-scale cues like light, for example, from the sun compass or electromagnetic field. You have also the wind direction that is given by the capillary waves in the surface. So what are you using? So to understand a little bit more what is happening, we developed this instrument. This was like an invention because when you try to put yourself in the place of the larvae, you have to be inventive. And this is a disc instrument which is completely transparent, and when it emerges in the ocean, and you have, it's basically a drifter with a environmental chamber, and this chamber is like this. It can be different sizes, and inside you put a larvae, and you look at the movement of this larvae inside the chamber. In this animation, you can see the compass is rotating. That means that the chamber has a drag underneath, and with the turbulence of the current is moving, is rotating. When you correct for the compass rotation, and here you can see the compass is always pointing to north. The sun is always at the same position, and the position of the larvae is now direction is towards a mean bearing. So you have accumulation of the position of the larvae within a direction, and you can measure that. Not only measure the mean bearing, but also the variability around this mean bearing, which in fact is a directionality. If you have higher variability, you have less directionality. So we tested this orientation factor with different, we test the orientation of larvae, again in the Mediterranean with, this is Villefranche-sur-Mer, with different factors like current speed, distance from shore, wind speed, sun elevation and azimuth, and cloud cover. So in short, only the celestial cues were influencing the directionality. The time of the day, for example, the sun, when it rises at the east, is a better signal than during the day, which is at the zenith. So the directionality decreased throughout the day, and also with the cloud cover, meaning that the sun is really an important cue. And that is critical because the sun is everywhere. So a larvae can see the sun from anywhere in the ocean, and if it can orient with the sun, then it will find the settlement habitat. We have tested this orientation towards the sun or away from the sun or some angle of the sun for these 4 different species, 3 of these different species have significant orientation to the sun. So this is really important. This is a finding that can be implemented now in the modeling. So I'm talking a little bit about the acoustic here. I don't know if we have sound. No, perhaps not. I don't think the sound works here. But we tested the acoustic cue because it was in the, like 30 years ago. Many of the ecologists, including Steve Simpson, assumed that the sound was a very important cue for these pelagic larvae. Actually, they are not. They are important cues for larvae that are coming near shore and to find a better settlement habitat. But from the offshore, we put the hydrophone, we deployed the disk with a hydrophone, we put some larvae inside, and what we find is that the larvae actually were emitting sound. And that was a discovery because we had no idea they could do this. Why would they do this? Well, what we did find is they vocalize only during the night. And you saw those larvae that are swimming together in groups. So we hypothesize that sun may enable larvae to maintain group cohesion at night. So we have deployed this chamber, some colleagues also independently in different places of the world. So we did a meta-analysis that is now that is in preparation by Egal Berenstein in the lab. And from 5 locations, we looked at the deviation of the previous heading versus the heading, which is telling us whether those larvae have external or internal cues. So basically, if they have external cues, is what I'm talking about with the sun compass. It could be a magnetic compass, something else. With internal cues, it's kind of like the gyroscopic kind of behavior where you can be directional for some time, and then you change the direction. So this could be the case because we did follow this larvae in the disk instrument only for 10 minutes. So, but by looking at the detail of their movement inside the chamber, we would be able to distinguish this. What we find is most of these larvae, except a few, like 2 species, have a bias correlated random walk. Bias correlated random walk is, I'm not going to go into the detail of that, but basically is following an external cues. The blue line is correlate random walk, and is that what we use in most models are using correlated random walk or simple random walk, like in the pink. Irregular would be those larvae that are bimodal, and there is a few species that are doing that. But basically, we know now that if we want to model these larvae, we have to use bias correlated random walk. And if we do that, we did that in the Florida Keys. Is also some work in preparation where we model different orientation to the sun, plus always adding the ontogenetic vertical migration. So we start from passive. This is a dispersal kernel for 30 days on top and 60 days on the bottom. So we're looking at the probability of settlement with distance. This is passive. Now we're adding ontogenetic vertical migration, which is observed. And we see that it increases the settlement at the source, decreases the dispersal distance by half. Now we're adding sound, and the sound doesn't change with ontogenetic vertical migration. It doesn't change much, but it does increase in terms of the distance, but it increases the settlement probability. And when we add the solar compass with OVM, only no sound, we find that this is really dramatically different. So sun compass is something that decreases the dispersal distance and increase the settlement. So what do we know now? We know that they migrate vertically during ontogeny, they settle to specific habitats, they swim fast, swim early, swim far, They can smell the reef. Also, I didn't show that, but they can. They can hear the reef. They can talk to each other, and they orient with sun compass. So they also have magnetic compass, and so they're not passive. And this is what the emerging paradigm is of larval migration rather than larval dispersal. We have to think about this aspect. So the aberrant drift hypothesis was tested and nuanced by the work in the Mediterranean by Robin Faitas and colleagues, and also by the modeling work with orientation behavior. That was a missing piece of your critical period hypothesis. So basically, when we revisit this hypothesis, which remember that it was during the first feeding stage, now we say that the settlement stage could be a critical hypothesis. And the reason for that is because if they lose the cues or lose the signal at sea, then they wouldn't be able to orient in the correct direction, and that would decrease the settlement. So what are, and I'm moving to the second part of the talk, where I'm really looking at, from what we know, what are the threats, and the work on fish larvae and the modeling of the transport brought to me to work with oil spill. Because at that time of the Deepwater Horizon, we were wanting to model what was going on like 1,500 meters below the sea. And here you see the oil spill footage just at the riser pipe when they cut the riser. And so this is oil mixed with gas that is rising into the water column. What is oil toxicity does in fish? This is a work by my colleague Martin Grossel and his team. They looked at mahi-mahi and looked at one of the most toxic components of the complex molecule of hydrocarbon, the polycyclic aromatic carbon. And what you see here is that it's really like a multi-system, multi-organ syndrome in the fish. When you quantify the oil impact on visual acuity of larval mahi using the optometric measurement here, where they have to follow this black and white line, when they're exposed to oil, you see that the visual acuity, even like with concentration of oil that are very low, like less to lethal, the lethal would be like 1 microgram to 2 microgram for this larvae, like 50% of the population. But here is less than 1, but the visual acuity decreases. And this is important just because I told you that the sun compass is important for this larvae. So they have very large eyes. They use this eye for many different things, including avoiding predation, but also orientation. So one thing that I want to point out is not only the oil we have to worry about, is subsea dispersion injection. So one of the response during the Deepwater Horizon was to use chemical dispersant, and that brings me back to Rachel Carson idea of like the DDT being really deleterious for the environment and for human. Here we are set to use this toxic chemical dispersant. It's not toxic, it's toxic by itself, but it increases the pH of the oil and suppresses the activity of biodegrading bacteria, and it remains in the environment for a long time. The supposedly advantage of it, and it's true, it decreases the interfacial tension of the drop of the oil between the oil and the water. That means that it decreases the droplet size, and by decreasing the droplet size, it decreases the buoyancy, basically, of this oil plume. And so there will be less oil rising to the surface, but more oil at depth. And that could be a trade-off. However, we recently did a study looking at the effect of this dispersion based on a large number of data that was released by BP on water column chemistry. And what we see, I'm not going to describe in detail here, but basically the message is that the subsea dispersion didn't change the plume that was formed at depth. You can see these red dots. This is a time series in the bottom of the PAH in the water column, and you have two layers that are high in concentration of pH, the surface layer and the bottom layer at 1,500 meters. The dotted line is when the wellhead was capped, and what we see is that there's still oil in the water column many months after the well was capped. And again, the dispersant was injected at different volume, and it didn't change anything statistically in the distribution of the oil in the water column. So there's not really any trade-off is using this. And why is that? Because we saw that it decreased the interfacial tension, it could decrease the oil droplet. Well, there are different processes that happen in the deep sea, and one of them is related to the fact that oil is not just crude oil, but is a gas-saturated oil component, and the gas under pressure is the solubility of the oil under pressure, the gas under pressure decreases, and also if you have a strong difference of pressure, the gas that compresses can expand rapidly, and that's what happened in the Deepwater Horizon. There was an 80 bar difference of pressure in the BOP, the blowout preventer, and the gas exploded and atomized the droplet. So when the injection of dispersant occur, the droplet was already small enough that the size was not changed. There's other factors like free gas, and I'm not going to talk more about this, but basically this is one of the danger that we see that may be affecting those pelagic environment. We developed, like I say, modeling for the Deepwater Horizon, and we use this model that was validated with the Deepwater Horizon data to model. This is an oil spill in the west part of Cuba. What you see here is like the three-dimensionality of this oil spill, where you have isosurface that you can select from different ppb or oil concentration. So you have higher oil concentration with the orange and the black at depth. And then at the surface, you see the gradient of concentration. So you have basically the mapping of the oil in 3 dimensions at all time that can be used to look at how this would affect the water column and the plankton and the fish larvae. So I want to end by looking at the revisited hypothesis from 100 years ago. We have dispersal hypothesis where the oceanography and the spawning run of the adult were driving the variability in fishery recruitment. And then you had those feeding hypotheses with a critical period, predation hypothesis. I didn't talk much about it, bigger and the better, mass mismatch. But I think we wanted to add the migration hypothesis, which is the oceanography with the larval dispersal behavior, and climate pollution hypothesis, see how this would change not only the mortality, but also the dispersal. Where we see right now is, for example, the embryo of mahi-mahi that are exposed to oil have different buoyancy. If we input the buoyancy of those embryos in the model, they will be dispersed in different strata in the water column, so they will be moved to different areas. and perhaps we have a settlement critical period. So these are all new ideas that are being developed to understand better the baseline of our population connectivity. So I want to say that as a scientist, we have confidence that we know the truth, and I think Rachel Carson understood this and had the courage to communicate it. She was an eye-opener, and Kofi Annan, who I know you all know who he is, used to live from his country tribal quote, says, gather these 5 virtues and you are a man: dignity, confidence, courage, compassion, faith. I think Rachel Carson had all these, and she was a woman. Thank you.
Thank you very much. We do have time for questions.
Yes, right. Yes, Raina?
So I'm wondering how changes in temperature, small changes in temperature, are affecting the settlement of larvae. What is known these days, maybe about or
So that's an excellent question. The question is from Rana Fine, how temperature may change the settlement of this larvae, small change of temperature. Well, I didn't focus much on that because I focus on the behavior more, but it's a really good question because we know that the PLD, which is a pelagic larval duration, depends on the growth rate of this larvae, which is directly linked to temperature. So this is another factor that also drive the settlement. And obviously with now changing climate and temperature, we can actually model that. We have developed some module to look at temperature taken from the model, because the model does not only have velocity, but it has temperature and salinity that can be used for the transport of this larvae.
Yes.
I'm Dave Jackson, seismologist at UCLA, and I just wanted to comment that this was a brilliant lecture, and I think the AGU should be really proud to bring together communities like this, and if you can communicate in a way that's really direct, and somebody from outside the field, I knew nothing about this. Your logical steps and the reason for your conclusions, and a picture of what's happening was really very well delivered. And I don't know if any of you go to seismology lectures and you ever get the same feeling. I hope so. But have you gone to a seismology lecture and found something that was inspiring to you?
I was not in a seismology lecture, but I was earlier in, I think Volcano from the Galapagos, and that was very inspiring to me too. So thank you for your comment. I really appreciate it.
And I should say thank you. Very well said.
Yes.
Hi, I'm Alex Phillips from Caltech. I'm just curious, what is the mechanism of the pH toxicity in the mahi larvae, and then what are some other suitable alternatives for bioremediation of oil spills that don't seem to affect it?
Thank you for your question about the pH mechanism in those fish larvae. I'm not doing this research. I'm not a toxicologist, but I work with Martin Grossel and his team to look at the effect of the changes of buoyancy, for example, of the embryos into the model. The pH they have like a paper in review now that lists all the different level of pH that affect different function of the larvae. They look at not only the behavioral function, but also the physiological functions, the heart rate and heart deformation. So it has like a range of different effects on the larvae and as well on the adult. And all this is really being taken into account with precision and model to understand the effect. We're also looking at spawning, the spawning location of these fish and, with respect to the deepwater horizon spill and how it may have affect the population. I'm sure I didn't answer your question, but these are questions more for the toxicologist. Yes, that's a very good point. Thank you. Yeah, when you look at climate and pollution, there's definitely adaptation. As far as there is no lethal direct impact, there is always fast adaptation, and we see that when we look at the genetics and fast evolving genes.
I think we need to close this now, but again, let's thank Claire for a really very nice lecture.